Oftentimes, a student's main job is only to learn in a familiar formula. Here's something new. Learn about it, study it, and take a test on it. In STEM classes, it's unfortunately missing the part where you get to actually participate in advancing science. That is, with the exception of Cornell iGEM, of course. Here, we are the scientists and the engineers, and we do exactly the things we learn about. Today, you're going to hear from a couple of our wet lab experts and all about how they do what they do. Quick note, if this is your first episode and you don't have much experience with biology or DNA specifically, I recommend that you start with our first episode, where I go through all the things you'll need to know for the podcast. All right, let's get into the lab. Well, before anyone actually gets into the lab, there's a couple things we have to get straight. First, what is it that an iGEM team does anyways? Here's Stan Cho to introduce us to that.
So synthetic biology is basically this concept of using cells to carry out some certain function that we want them to. And the way that we alter a cell's function is through gene editing, which is this process of changing the DNA sequence that you can find inside a cell. What this does is then changes what types of proteins are either made or knocked out inside of a cell, which effectively changes its function. And so once you have synthetic biology in mind, we can then address certain problems that we can identify. And then when we look into these specific problems, we can understand the pathways that are taken to create certain proteins. Once these pathways have been identified, then we can integrate that into a design where we are actually using gene editing to come up with the results that we want. And then we can integrate these and execute them with certain cells that have these gene-edited DNA in them.
The second thing then is how do we know what we're going to be putting in the bacteria to get what we want? Here's Arya Chinay, one of our wet lab colleagues this year, to walk us through the planning stage.
So this is where we get into plasmid design. Designing a plasmid is essentially like building with Lego bricks, but in iGEM we call them biobricks, and they're these really well-characterized DNA sequences that each have a specific function, and they can be assembled in a modular way. So when I design a plasmid, I like to take it step by step. It all starts with a promoter, which is the on switch for transcription. This determines how much and when our gene is expressed in the cell. So a really strong promoter would produce a lot of RNA transcript for our gene. And then we can also have constitutive promoters that are always on and always transcribing the gene, or inducible promoters that require some kind of signal that turns on the promoter. Right after that is the ribosome binding site, or RBS, which controls how efficiently the protein gets translated. So even if two different plasmid constructs have the same promoter, they can have vastly different protein levels based on the strength of the RBS that they include. Next is the gene of interest. So this is the actual coding sequence for whatever function we're trying to introduce into the cell. This could be producing a protein or a fluorescent reporter like GFP, or some part of a larger pathway. Any other function that we're trying to use our model organism for, this is really the heart of it. Depending on the application, we might also include tags or signals onto our coding gene. For example, we could include a secretion tag if we want a protein to be exported out of the cell, or if we want to produce a protein and purify it, we could also include affinity tags like His tags. And we can also include degradation tags if we want to control the lifespan of our protein. After the gene, we include a terminator, which tells the cell when to stop transcription, and it can keep the cassette together. And for context, a cassette is a functional unit of DNA in our plasmid, so it's our coding gene plus all of the regulatory elements that I'm talking about, like the promoter and the terminator and the tags. But the plasmid isn't just this one expression cassette. We also need really important backbone elements that keep the plasmid functional inside the cell. One part of the backbone is the origin of replication, which determines how many copies of the plasmid are inside the cell. And the origin of replication is exactly what it sounds like. It's the place where replication of the plasmid starts. We also include a selection marker. This is super important, and usually this is an antibiotic resistance gene that helps us select for cells that actually took up our construct. And one big question is, where do these DNA sequences even come from? How do we get the ATCGs that we need to build this plasmid? So iGEM has a very handy website called the iGEM Registry. It's basically a collection of thousands of DNA sequences called biobricks that have been constructed by the iGEM community and all of these scientists and students that work on DNA sequences. And it's a really, really handy tool when you're thinking about your plasmid design, because it has so many parts from old iGEM projects or old studies that have found success in these parts. And they're all modularized, so you can assemble them without any heavy lifting on the DNA engineering.
Now we're ready to get into the lab. We know what we're looking for, so it's time to actually make it. The two most important things for us now are cloning and transformation. Let's hear a little bit about that from Stan again.
Cloning is the process of making identical copies of DNA sequences, and the reason why we would pursue cloning in synthetic biology projects is to copy our genetically engineered gene of interest. And once we copy a huge amount of these DNA sequences, we can then transform them into specific cells, typically bacteria like E. coli. And what these cells do is that they take these plasmid DNAs and then they are able to use them to generate the specific gene of interest that we coded into our DNA sequences.
It's worth drawing a clean line between these two related terms, since sometimes they blur together. Cloning is about the DNA itself, specifically building and copying a defined DNA construct, like inserting a gene into a plasmid and making copies of that sequence. Transformation is about the cell. Essentially, it's putting that plasmid into a cell. Let's get into the weeds a bit. First, let's talk about PCR, or polymerase chain reaction. Essentially, PCR is a molecular photocopying technique. Scientists take a fragment of DNA and make millions or billions of copies in a short amount of time. It hinges on replication in a controlled, repeatable cycle. It starts with heat, which causes the two DNA strands to unzip. Small pieces of DNA which you design, called primers, attach to the start and the end of a specific piece of the longer DNA strands you want to make copies of. This step has to happen at a specific, lower temperature that allows the enzyme, usually Taq polymerase, to build new DNA strands by adding the complementary nucleotides to each DNA strand. The most important part is the CR in PCR, as in chain reaction. Each cycle will double the amount of target DNA, so after about 30 cycles, you're working with over 1 billion copies of the specific gene you want. PCR is used all throughout synthetic biology, and it'll be mentioned again soon. Before I pass it off, let's mention 4 related terms that come up all the time when working with DNA: endonucleases, exonucleases, ligation, and overhangs. Nucleases in general are enzymes that cut DNA, but the way they cut DNA is different. Endonucleases cut within the DNA strand. Depending on the enzyme, those cuts aren't always right between 2 pairs of nucleotides. This is where overhangs come in. When some endonucleases cut DNA, they leave short, single-stranded ends. Sometimes they're referred to as sticky ends because they can just base pair with complementary sequences, like jigsaw puzzles. 2 overhangs made by the same enzyme will often be compatible, making them easy to join. The joining process is called ligation. It seals the DNA backbone and makes the connection permanent. Some endonucleases don't make overhangs though, they make blunt ends. These make it less efficient to connect 2 strands together because you don't have the base pairing to make it easier. How about exonucleases? Well, these enzymes don't cut in the middle, but they work at the ends of DNA strands to remove nucleotides. Instead of making fragments, they trim down the DNA. To talk a little bit more about cloning and transformation techniques which use endo and exonucleases, here's Christian Funkelsrud.
Alright, so there are multiple types of cloning that we use in the lab. One of the traditional ones that was originally used in molecular biology is restriction enzyme cloning. We've moved away from the traditional restriction enzyme cloning because that leaves scars in our recombinant DNA. So what that means is that with these endonucleases, the restriction enzymes, they have a restriction enzyme site that they recognize, and then they cleave at that site, and that leaves that scar of the site inside of our recombinant DNA. So with these new techniques, such as Gibson assembly or Golden Gate assembly, it creates scarless products, which is more what we want. So with Gibson assembly, the exonuclease chews back on the overhangs you create on your PCR products, and then that creates overhangs that can then come in contact with each other and ligate if they match up. And that if you design the overhangs correctly, or the PCR products, then you have scarless DNA. And then with Golden Gate assembly, you have type 2S restriction enzymes, and what they do is they're endonucleases that cleave outside of the restriction site, so they're able to just cut out the restriction site, leaving you with scarless recombinant DNA. There are other common cloning techniques such as topo assembly that are used, but in iGEM at Cornell, we mainly use Gibson assembly. For transformations, when we're doing E. coli transformations, we use heat shock, so we use chemically competent E. coli, and there's a protocol where we can just heat up the E. coli, which disrupts their cell wall, so the plasmid kind of flows inside of the E. coli. We plate them, and they're able to grow with our new recombinant plasmid. There are other techniques we use for higher efficiency transformations or in other organisms like yeast, where you do like lithium acetate heat shock transformations, or you can do electroporation, which is where you shock the organism with electricity, and the electricity disrupts the cell wall again, and then the plasmid's able to flow inside, and then when they recover, they have your recombinant plasmid inside. So with transformations, one thing you can do is just insert your plasmid inside of, say, a bacteria or yeast cell, and then have them expressing the gene of interest. Another thing that you can do is actually integrating that inside of the bacterial or eukaryotic cell's genome, and then have them constantly express that while it's in their genome, which is much more stable. So if you don't have a drug selection marker in your plasmid, then it can easily get spit out by whether it's a yeast cell or the bacterial cell that you're using. So what you can do is insert that inside of the organism's genome and then have them more stably express that construct.
Now that we've got all that sorted, we have another step. We have to make sure that what we made actually worked. Getting colonies isn't success. Getting the right colonies is. Here's Parth Patel to tell us all about our testing process.
So first, we check if the DNA we wanted is actually inside the cells. The way that we would do this is with a colony PCR. Essentially, you pick a colony and you amplify the region that we care about, and we see if we get the expected product. Then we run that DNA on a gel.
Pause. Gels are used for gel electrophoresis. Essentially, DNA carries a negative charge, so when you put it in a gel and apply an electric field, DNA fragments will move towards the positively charged end of the electric field. These fragments have to be prepared, usually through PCR, to get better readings. Since smaller fragments will move more easily, they'll end up further along the gel than longer and therefore heavier fragments. The groups of fragments will show up as bands, and we're able to figure out just how long these fragments are by how far they moved.
Gel electrophoresis is essentially our way of confirming the size of the fragment. If the band on the gel shows up in the right length, then that's a good sign that our insert was successful. To be more certain, we can sequence the DNA. This tells us the exact order of bases, so we know that the construct is correct and hasn't been mutated. But even if the DNA is correct, that's not the full story still. The real question is whether the system actually does what we want it to. So the next step is functional testing. Does it produce product? Does it fluoresce? Does it respond when we induce it? Basically, does the biology behave the way that we designed it to? That's how we go from just having cells to actually proving that we've successfully transformed.
Since we are Cornell iGEM, the greatest iGEM team on the planet, we get everything right the first time, and we don't have to worry about fixing our mistakes.
Oh, wait.
No, absolutely not.
No.
What is bro talking about?
That's not how that works.
Well, I guess even the greatest project teams don't always get it right the first time. Like every other engineering discipline, this is all part of a process. Design, build, and test. When we discover something has gone wrong at any of these steps, we restart the process. Back to the scratchboard. Let's figure out what went wrong. Here's Sylvia Park, our other wet lab co-lead, to go through the process of iterating and troubleshooting.
So when we have failures, the way we go about it is we usually try to locate what the source of the failure is. So for example, last year in our Project Prosper, we had trouble with the transformation, and we got successful colonies with the plasmid that we wanted to transform it with, but we realized that the plasmid had not been properly ligated with our insert. So what we had to do to figure this out is we had to sequence our transformed bacteria to see that the sequence of the plasmid inserted did not have the insert. The way we go about trying to figure out what went wrong in a project is we usually try to insert more checkpoints during the project. For example, running gels, running nanodrops, sequencing. All these different mechanisms involved help with ensuring that everything goes right and we catch failures as we go instead of trying to troubleshoot and figure out what went wrong during our experiment.
We've covered a lot of ground today, yet we've only touched the surface of the amazing things we do here at Cornell iGEM. Feel free to leave a comment if you're curious about anything you heard today, and if you enjoyed this new style of teaching, make sure to follow or subscribe. This is the end of this episode, but it's certainly not the last you'll hear about this. We're going to go deeper into each of these sections: DNA sequencing, model organisms, techniques, and more. There's a lot waiting for you, so stay smart, and I'll see you geniuses soon.